Hello, my stunning students. I hope you're ready because it's another biology with Miss Judd. So we just wrapped up mitosis, and at the end of mitosis, we created two identical cells, which is really important and awesome because they're used to replace old cells, it's used to heal, and it's a really important process. Today, we're going to dive into another type of cell division called meiosis. Sounds super similar. Meiosis is a special form of cell division that occurs in organisms that reproduce sexually. So typically, eukaryotic organisms are reproducing sexually, like our plants and our animals. And I'm going to send it to Ghost Miss Judd to talk about why this process is important.
If we did not go through this meiosis process, then the male and female reproductive cells would both have 46 chromosomes, which through fertilization, if you were to add 46 and 46, then that zygote, that embryo, would have 92 chromosomes, or double the number necessary. So we need to split that number in half before fertilization. And that's where meiosis takes its place. So we reduce the number of chromosomes in half. So we start off as an adult human with 2N, or 46 chromosomes, 2 sets, and this process of meiosis creates one single set, or haploid, the amount of chromosomes, so that by fertilization, the resultant zygote is your total number of chromosomes.
So this process of meiosis is taking a single cell and it goes through 2 cell divisions. At the end of this process of meiosis, I end up with 4 cells. They have half the genetic information and they are completely different. This allows us to have some genetic variation, which means you don't look like your siblings, which means you don't look like your parents, and that's awesome because that means that our world is unique and colorful and full of so many different types of organisms. We are about to jump into our guided notes, so make sure you have your notes next to you as you're watching this video. You are going to handwrite. Remember, we want to use our brains efficiently. And I'm going to send it up to Miss Judd in the corner. Hi, guys. Miss Judd in the corner here to guide us through Unit 2, Lesson 3, Meiosis, which does sound a lot like mitosis, and it kind of is similar. So we're going to pull on a lot of our mitosis knowledge here. In meiosis, we are forming our gametes. So the whole purpose of this, it's a form of cell division to produce our sex cells, gametes, the egg and the sperm, in order to go through sexual reproduction. So any organism that goes through sexual reproduction, like plants, like animals, go through this process of meiosis. The goal of this process is to create a haploid cell. So we're moving from a diploid cell to a haploid cell. In order to do this, we go through 2 divisions, meiosis 1 and meiosis 2. Now we've just learned 2 new words, haploid and diploid. Miss Judd, what do those mean? Never fear. Haploid and diploid. Now, our body cells and every organism's body cells are diploid. There are 2 copies of each chromosome. One chromosome comes from mom. The other chromosome comes from dad. So we can see they go together. They contain the same information, but you get one from mom, one from dad. In a haploid cell, you only have 1 copy. So if you look, we only have 1 set. We are trying to move from this diploid cell into a haploid cell. Before any cell division, we have to go through interphase, which means we go through G1, S, and G2. In S, chromosomes are replicated, so we have 1 sister chromatid, and it becomes 2 sister chromatids. Each duplicated chromosome consists of those identical chromatids attached at the centromere in the center. Once we go through interphase, we can start our cell division with meiosis 1, and each division goes through that PMAT from mitosis. We start off with prophase 1. In this case, my chromosomes condense, they appear, and they pair up. So prophase 1, pair up. They pair up to form a tetrad because we have 4 chromatids in a tetrad. In this case, crossing over occurs, which increases our genetic variation. And in part 2, we're going to talk about genetic variation. If you look in crossing over, some of the red goes on the blue, some of the blue goes on the red, and we are sharing information so that these two homologous chromosomes are no longer identical. After prophase, we move into metaphase 1. In this case, the tetrads, those 4 chromatids, those pairs, line up in the middle. This is random. That's called independent assortment when it's just randomly lining up in the middle, which again causes more genetic variation. After metaphase 1, we move into anaphase 1. Things are getting pulled apart. And in this case, it's really important to note that in anaphase 1, my homologous chromosomes are getting pulled apart. My sister chromatids stay together, so the whole X pulls us apart. After anaphase is telophase. So we have telophase 1, where now we have 2 new nuclei. But in each of these, my chromosomes are no longer identical, and we've separated those sets. So now we are haploid. Now we are haploid cells. I only have 1 big chromosome and 1 small chromosome on each side. No longer 2 sets, we are back to 1. Cytokinesis occurs, and 2 non-identical haploid daughter cells are formed. Now we can start meiosis 2. Meiosis 2 starts with prophase 2. In this case, again, the DNA is condensing. Chromosomes become visible. My nucleus disappears. My spindle forms. This should sound a lot like mitosis because it is. After prophase, we move into metaphase. In metaphase 2, my chromosomes line up across the center of the cell in a single file line, different from metaphase 1. My spindle attaches. After metaphase, we have anaphase. In anaphase 2, the copied chromosomes move to the opposite ends of the cell. So my sister chromatids separate. And this should look like mitosis, what we already know. After anaphase, we have telophase. Now in telophase 2, we have 4 nuclei forming. The chromosomes uncoil, the spindle disappears, and my end goal of meiosis, what I end up with is 4 haploid unique daughter cells that are called gametes. The female gametes are egg, the male gametes are sperm. Cytokinesis occurs to get those 4 cells. All right, part 2. What is this genetic variation we're talking about, and why is it so important? Our genetic variation happens in 2 places of meiosis. First of all, in prophase 1, crossing over. My homologous pairs join together in that tetrad and exchange information and alleles so that my chromatids are no longer exact replicates. If we look, we have switched information through this process of crossing over. During metaphase 1, the chromosomes line up in the center randomly, which is known as independent assortment. So looking at this picture, we have a random different ways these chromosomes could line up in the center, which at the end of meiosis leads to completely different cells. This genetic variation is so important because that is how we all look so different. This is how we as individuals have unique traits, and we don't look exactly like our parents or our siblings. All right, so let's compare them all. At the bottom of the page, I want you to compare mitosis to meiosis. Good luck, you guys. Thank you, Ms. Judd in the corner. On your checklist, there's a link to a video. The Amoeba Sisters of Meiosis is absolutely spectacular. So if you want to hear this again, if you want an extra resource just to make sure you understand this process, please go check out that link. Now that your notes are handwritten, we can just submit those to Seesaw. Remember to take a picture and upload that. We are going to take our lesson check for the day, and then that's it. That's all you got. Remember, you guys are great learners. You're doing such good work, and I am so proud of you. Have a good day and make good choices.